Good morning, students. Myself, Dr. Ragasudha, Head and Assistant Professor, Department of Microbiology, Government City College, is here with a topic on vaccines. A vaccine is a biological preparation that improves the immunity to a particular disease. A vaccine typically contains an agent that resembles a disease causing microorganism and is often made from weakened or killed form of the microbe. The agent stimulates the body's immune system to recognize the agent as foreign, destroy it, and keep a record of it, so that the immune system can more easily recognize and destroy any of these microorganisms that it later encounters. The term vaccine and vaccination are derived from variole vaccinae, a virus named for smallpox of the cow. The term devised by Edward Jenner to denote cowpox. History. Jenner used cowpox pustules to immunize against smallpox in 1796. In 1800, William B. Coley tested the idea that the immune system can be reprogrammed to fight cancer. In 1801, 1 lakh people were vaccinated in Europe. President Thomas Jefferson and Harvard professor B. Waterhouse spearhead US vaccination campaign. In 1821, Britain passes compulsory vaccination laws opposed by working class Britons. In 1879, Louis Pasteur has developed a vaccine against cholera. In 1885, Pasteur demonstrated the use of his attenuated virus rabies vaccine. In 1890, tetanus and diphtheria vaccines were developed. In 1916, first large polio epidemic in the US occurred. In 1935, first field trials for polio were conducted. 20,000 children were vaccinated, 9 died. Media plays close attention. In 1954, field trials for Salk vaccine began. 1.8 million children eventually involved. In 1961, Sabin vaccine was licensed. Kennedy makes vaccinations a key issue for his administration. In 1962, oral polio vaccine was developed. In 1967, 26 number of vaccine manufacturers in the US have established. In 1974, MMR vaccine causes controversy in UK linked to autism in children, vaccine rates decline. From 1977 to 79, 90% of vaccine manufacturers drop out of the marketplace in the late 1970s. 2000, during 2000, Prevnar became the first vaccine to generate more than $1 billion in sales. President George Bush receives a smallpox vaccine as part of public health campaign in 2002. In 2003, Bill and Melinda Gates launch Grand Challenges in Global Health Initiative focusing on the creation and improvement of vaccines. Now, 5 of major US firms in the vaccine market have developed in 2006. In the same year, 3 new vaccines have launched: Gardasil, Rotatec, and Justavax, the most ever recorded in a single year by Merck. Merck records revenue of $2 billion in the first half of 2007 from vaccine sales. So, this is a brief history regarding the development of vaccines in the world. Now let's see the development of vaccine in India. In 1893, efficacy trials on cholera vaccine conducted in Agra, India. 1897, first plague vaccine developed by Dr. Hafkin. In 1904 to 5, first vaccine research institute was established at Kasauli, Himachal Pradesh. In 1907, Pasteur Institute of India, Kunur, manufactured neural tissue anti-rabies vaccine. In 1920 to 39, DPT, DT and TT vaccine became available in the country. In 1940, Drug and Cosmetic Act enacted. In 1948, BCG vaccine laboratory was set up in Gundi near Madras. In 1951, liquid BCG vaccine became available in India as part of mass campaigns. 1965, live attenuated freeze-dried smallpox vaccine became available. In 1967, freeze-dried BCG vaccine became available. OPV became available in India. In 1970, the first time in India, indigenous oral polio vaccine trivalent called Sabin vaccine was developed and produced. In 80s, indigenous measles vaccine production started. In 84, inactivated polio vaccine first produced in India. In 85 to 88, AEFI surveillance system established and initial guidelines were released. In 89, Indian Vaccine Company Limited and Bharat Immunologicals and Biological Limited were set up as public-private joint venture companies. In 97, first ever recombinant DNA hepatitis B vaccine was developed in India. In 2006, guidelines for climate trials by Indian Council of Medical Research was established. In 2009, three Indian manufacturers developed a pandemic flu novel H1N1 2009 vaccine. In 2010, National Pharmacovigilance Program of India launched measles A vaccine for African meningitis, belt licensed and successfully used in campaigns in Africa. Indigenously researched bivalent oral cholera vaccine developed and licensed in the country. In 2012, an indigenous inactivated JE vaccine licensed in the country. Indian manufacturers acquired a capacity to produce inactive polio vaccine. Vaccination milestones in India. In 85, UIP launched with 6 antigens BCG, DPT, OPV, and measles. In 86 to 2001, no new antigen is added to the UIP of India. From 2002 to 3, hepatitis B vaccine became the part of UIP in 14 metropolitan areas and 33 districts of the country. 2006 to 10, Japanese encephalitis vaccine added to the UIP in select endemic districts in phases manner. JE vaccination is initially done through a mass campaign to cover 1 to 15 years of age group, followed by administration in routine immunization schedule at an age of 16 to 24 months. 2007 to 8, hepatitis B vaccine scaled up to cover 10 additional states of India. 2010, second dose of measles vaccine introduced in UIP in India. 21 states introduced vaccines in UIP at the time of DPT booster 1, and the rest 14 states started measles catch campaign to cover 9 months to 9 years of age children. Japanese encephalitis vaccine campaigns completed in all identified 112 endemic districts of 15 states of India. JE vaccine became part of UIP in these districts in 2010. In 2011, measles catch-up campaign continued. Hepatitis B vaccine scaled up to UIP of all 35 states and UTs in India. Haemophilus influenza type B (HIB) vaccine introduced as pentavalent along with DPT and hepatitis B vaccine in UIP of Tamil Nadu, Kerala states of India. In 2012, measles catch-up campaign completed in 9 states of India and ongoing in remaining districts of 5 states of India. The plan for expansion of pentavalent vaccine in 6 additional states and UTs of India. Vaccines production in India. There are a number of licensed vaccines manufacturers in India. These manufacturers have installed capacity and are licensed for production or marketing of at least one or more vaccines in India. They are BCG Vaccine Laboratories, Gundi, Institute of India Limited, Pune, Pasteur Institute of India, Kunur, Central Research Laboratory, Kasoli, Hafikans Biological Production C Limited, Mumbai, Human Biologicals and Immunologicals, Hyderabad, King Institute of Preventive Medicine, Chennai, Pasteur Institute, Shillong, Biovaccines, Hyderabad, Dano Vaccines, Hyderabad, Bharat Immunologicals and Biological Company, Bulandshahr, Pancare Biotech Limited, Delhi, Biomed P, Ghaziabad, Bharat Biotech International, Hyderabad, Sanofi Pasteur India Private Limited, Delhi, Zydus Cadila, Ahmedabad, Chiron Behring Vaccine Lab, Akleshwar, Gujarat, Sanofi Pasteur, New Delhi, etc. Now let's see the types of vaccines. There are 7 types of vaccines: live attenuated vaccines, inactivated vaccines, subunit vaccines, toxoid vaccines, conjugate vaccines, DNA vaccines, and recombinant vector vaccines. Live attenuated vaccines contain a version of living microbe that has been weakened in the lab so it cannot cause disease. Because a live attenuated vaccine is the closest thing to a natural infection, these vaccines are good teachers of the immune system. Example, vaccines against measles, mumps, and chickenpox. Inactivated vaccines. Scientists produced inactivated vaccines by killing the disease causing microbe with chemicals, heat, or radiation. Such vaccines are more stable and safer than live vaccines because dead microbes cannot mutate back to their disease causing state. Example, vaccine against influenza, polio, hepatitis A, and rabies. Subunits vaccines. Instead of the entire microbe, subunit vaccine include only the antigen that best stimulates the immune system. In some cases, these vaccines use epitopes, the very specific part of the antigen that antibodies or T cells recognize and bind to. Because subunit vaccines contain only the essential antigen and not all the other molecules that make up the microbe. Example, plague immunization. Toxoid vaccines. For bacteria, the secret toxins or harmful chemicals or toxoid vaccine might be the answer. These vaccines are used when a bacterial toxin is the main cause of the illness. Scientists have found that they can inactivate toxins by treating them with formalin. Such detoxified toxins called toxoids are safe for use in vaccine. Example, Carotolus atrox toxoid is used to vaccinate dogs against rattlesnake bites. Conjugate vaccines. If a bacterium possesses an outer coat of sugar molecule called polysaccharides, as many harmful bacteria do, researchers may try making a conjugate vaccine of it. Polysaccharide coatings disguise a bacteria's antigen so that the immature immune system of the infants and younger children cannot recognize or respond to them. Example, Haemophilus influenza type B vaccine. DNA vaccines. Still in the experimental stages, these vaccines show great promise and several types are being tested in humans. DNA vaccines take immunization to a new technological level. These vaccines dispense with both the whole organism and its parts and get right down to the essentials, the microbe's genetic material. Example, influenza vaccine. Recombinant vector vaccines. Recombinant vector vaccines are experimental vaccines similar to DNA vaccines, but They use an attenuated virus or bacterium to introduce microbial DNA to cells of the body. Vectors refer to the virus or bacterium used as the carrier. Example, DPT vaccine. Saponins as vaccine adjuvants. At first, what is the vaccine adjuvant? A vaccine adjuvant is a substance that is added to the vaccine to increase the body's immune response to the vaccine. Saponins are natural glycosides of steroids or triterpenes, which exhibit many different biological and pharmacological activities. Notably, saponins can activate the mammalian immune system, which has led to significant interest in their potential as vaccine adjuvants. The most widely used sapon-based adjuvants are Quil-A and its derivatives QS21, isolated from the bark of Quilingi saponaria molina, which has been evaluated in numerous clinical trials. This unique capacity to stimulate both the Th1 immune response and the production of cytotoxic T lymphocytes against exogenous antigens make them ideal for the use in the subunit vaccines and vaccines directed against intracellular pathogens as well as for the therapeutic cancer vaccines. Research and development in vaccines. WHO has a separate unit for the research and development of vaccines. WHO Initiative for Vaccine Research facilitates vaccine research and development against pathogens with significant disease and economic burden, with a particular focus on the low and middle income countries. Finding a safe, effective, and durable HIV vaccine remains a top priority for the world. Through the Vaccine Research Center and the Division of Acquired Immunodeficiency Syndrome, NIAID is recently busy in conducting and supporting biomedical research that leads to increased knowledge about how HIV interacts with the human system and evaluation of the most promising vaccine candidates. Now let us see how this vaccine works. Vaccine contains a modified form of virus or bacteria that does not cause disease but does teach your immune system what to do if you are ever attacked by the real potentially dangerous virus or bacteria. When you get vaccinated, your immune system responds just as it does to any other intrusion by creating antibodies to fight off the particular virus or bacteria. After vaccination, your body remembers this specific intruder. If you ever come in contact with the real virus or bacteria, the right antibodies quickly destroy it before it has a chance to make you sick. For some diseases, more than one dose of the vaccine or a booster dose later in life may be needed to ensure full and lasting protection. Now let us see what is herd immunity. Before vaccines, measles is one of the most contagious disease affecting humans. Before vaccines were available, there was no way to stop the disease from spreading. Each time a community was exposed to the virus, nearly everyone who had not been infected before contracted the disease. Many suffered serious complications such as pneumonia, deafness, diarrhea, and may death. Building the herd. Measles moves quickly. The higher the number of the vulnerable individuals there are in a community, the higher each person's individual risk will be of contracting the disease. As more and more people are vaccinated, it became more difficult for the virus to find someone new to infect. Protection for all. Thanks to the measles vaccine, the number of measles cases in WHO Europe region has dropped over 90% since 90s, but the virus still spreads whenever it can find enough people to infect. By stopping the chain of infection, Vaccinated children and adults prevent the virus from potentially reaching babies too young to be vaccinated and people who cannot be vaccinated because of weakened or impaired immune systems. At least 95% immunization coverage is needed to achieve the herd immunity protection for all. So, this is a good example for developing a herd immunity. The same thing is done with respect to any disease which spreads very quickly in the community. Stages of vaccine development, it has 5 stages. The first one is preclinical stage. Second one, phase 1 safety trials. Phase 2 expanded trials. Phase 3 efficacy trials. And the final stage is approval stage. In preclinical stage or preclinical testing, vaccine is given to animals such as mice and monkeys to see if it produces any immune response. In phase 1, tested on a small number of people for safety and dosage and to confirm that it stimulates the human immune system. In phase 2, vaccine is given to hundreds of people split into groups by age. Phase 3, testing expanded to thousands of people. And final stage is the approval phase where regulator in each country review the trial results and decide whether to approve the vaccine or not. As we are in COVID-19 pandemic situation, here it's very important to compare how the classical vaccines were developed and the current COVID-19 vaccines are being developed. Classical vaccine preclinical stage took 18 to 30 months, whereas in COVID-19 it's completely absent. Phase 1, dozens of voluntaries uh, up to 30 months. Here, dozens of voluntaries only to 6 months. Phase 2, hundreds of voluntaries up to 32 months. Again, here 6 months. And phase 3, thousands of voluntaries up to 30 months. And the final approval and manufacture and vaccination took approximately 12 to 24 months, whereas here there is no phase 3 directly. The vaccine is for approval, manufacture and vaccination. Billions of doses or individuals are and it takes about 6 months. So this is how the classical vaccines were developed and the current COVID-19 vaccines were being developed. Tracking COVID-19 vaccine. Here we can see some companies, sponsors or institutes which are developing COVID-19 vaccine. The University of Oxford, it has been approved for vaccine manufacture. Moderna is also been approved. Pfizer and BioNTech is also been approved for vaccine manufacture. Wuhan Institute of Biological Products, China, and National Pharmacological Groups is also in the race for vaccine approval. Beijing Institute of Biological Products, China National Pharmaceutical Groups is another company from China which is for development of the vaccine. CanSino Biologicals, Inovio Pharmaceuticals, then Sinovac, Novavac, CureVac, Imperial College London, Johnson and Johnson are the other companies which are in the race for development of vaccine, COVID-19 vaccine manufacture. Now let us see vaccines which are to be given to the children. BCG vaccine for TB and bladder cancer, hepatitis B vaccine for hepatitis B, polio vaccine for polio, DTP is for diphtheria, tetanus and pertussis, HIB is for infection caused by bacteria, PCV for pneumonia, RV for severe diarrhea diseases, typhoid for typhoid fever and diarrhea, MMR is for measles, mumps and rubella, varicella for chickenpox, Hepatitis A is for liver disease and TDAP is for diphtheria, tetanus and pertussis. So the minimum age for dose 1 for BCG is can be for 5 years. Hepatitis B is for 1 month. First dose is at 1 month and the second and third doses can be given at 5 months. Polio virus or polio vaccine is given first month after birth and later for 6 months. After every 6 months it can be given. DTP is given anytime. The first dose is usually in the first month, immediately after the baby is born, and the second dose is in the fifth month. Hib vaccine is given in the fourth week of the birth of the baby. Then in sixth month, booster dose is given. Another booster dose can be given after another 6 months. Then PCV dose is given to age between 2 to 5 years of age because this pneumonia is found for kids between the age group of 2 to 5. Similarly, RV is given in the fourth week of baby born. Then typhoid can be given at any time. MMR is given for school going age children. Varicella is given between 7 to 12 years. Then hepatitis A can be given again at any time, and Tdap is given from 7 to 10 age. Then let us see some adult immunization schedule by age group here. For adults, there are vaccines like influenza inactivated vaccine or influenza recombinant vaccine, then influenza live attenuated vaccine, then tetanus, diphtheria and pertussis vaccine. Tdap can be given for adults. MMR is also given for adults. Varicella is given for adults in 2 doses. Zoster recombinant is given for age group people from 50 to 65 years of age. Then human papilloma female, for females it is given in 2 doses. Then human papilloma for male is also given in 2 to 3 doses. Then pneumococcal conjugate is a single dose vaccine. Pneumococcal polysaccharide is again given in 2 doses, 1 or 2 doses. Hepatitis A 2 to 3 doses. Hepatitis B again 2 to 3 doses. Meningococcal ACWY is given in 1 or 2 doses, and even booster dose is required after 5 years if there is a risk. Meningococcal B 2 to 3 doses. Haemophilus influenza is given in 1 or 3 doses depending on the indication. Thank you.